Good afternoon. My name is Joelle Sarlls. I'm one of the staff scientists in the NIH MRI research facility in Building 10. And today I'm going to talk to you it's an introductory lecture on diffusion weighted imaging. And this is, of course, an education lecture, so feel free to ask questions as we go along. The whole point is to make sure that you get something out of it today and hopefully that you learn something. So the motivation for giving this talk today is that, as you know already, that magnetic resonance imaging provides information about the spatial distribution of water, so very basic structural MRI. In diffusion-weighted MRI, we get information about the motion of these water molecules that are in the tissue. And today I'm going to be focusing all on brain imaging, which I think has been the rest of the course also. And diffusion-weighted imaging is sensitive to the cellular architecture and potentially the tissue integrity. So this is one of the reasons why we want to look at diffusion images, is to be able to say something about this cellular architecture, the microstructure of the tissue. And diffusion-weighted imaging can provide quantitative measures that are directly comparable either between subjects or between different time points. So that's one of the benefits of having a quantitative measure. In addition, diffusion imaging can be used to identify specific bits of white matter and that you want to maybe investigate further or that you think might be different between populations. So here's an outline of my talk. I'm first going to describe what is diffusion. What are we talking about when we say diffusion imaging? How do we measure diffusion with MRI? How do we extract some directional information from this data? What are some of the practical problems and limitations of acquiring and using diffusion images for when you go and run these studies on your own? And then I'll also touch on beyond the diffusion tensor representation. So first, what is diffusion? Diffusion refers to the random translational or Brownian motion of molecules that results from the thermal energy of these molecules. And the diffusion coefficient is given by the Stokes-Einstein equation here, and it is directly proportional to the temperature and inversely proportional to the viscosity of the medium that the particle is diffusing through, and then also inversely proportional to the size, here represented as the radius of a sphere, of the particle. So if we have a large number of particles that are free to diffuse, then they have a square displacement of the Gaussian form. So this is where, if we have particles, a whole ensemble, and here I've got this one-dimensional representation. So if we take this whole ensemble and have them at the zero location, so zero displacement, and we let them start diffusing, then after some time, diffusion time here, then they will have displaced and spread out and have this sort of distribution, this Gaussian form, this bell curve. If we look at this in 2 dimensions now, this is like a cone shape, and then in 3 dimensions, this is a sphere. And so the displacement is given by the square root of 6 times the diffusion coefficient of the particle and the time that we've allowed for the diffusion. So more time, they will have diffused farther. Okay, so in this course, we're talking about diffusion in the MRI of living tissue, so in vivo MRI. So the particles we are talking about are water molecules. And water molecules at body temperature being 37 degrees Celsius, we know from other experiments that the diffusion coefficient is 3 times 10 to the negative 3 millimeters squared per second. So that's already a given. And if we let these water molecules, an ensemble of them, diffuse for 30 milliseconds, which is a relevant time for MRI, then they will have moved on average about 25 microns. So for those who are neuroscience based, then you'll know that this is at the length scale of cells and axons and whatnot in the brain. So this is for free water, that it will have moved about 25 microns. And if the motion of water is hindered or restricted by cell membranes and the myelin sheaths of axons, macromolecules, etc., then the displacement will be less than that of the free water, and so the diffusion coefficient we measure will appear lower. Okay, so Now we've got an idea about what diffusion is. How can we actually measure this with MRI? Recall that in MRI, the image intensity is governed by some physical properties of tissue water. So there's the proton density, the T1 relaxation time, the T2 relaxation time, the T2 star relaxation time. And so now to this, we're adding the physical property of the diffusion coefficient. And this tells us again about the translational motion of the water molecules. And to be able to make our image intensity more sensitive or less sensitive to these physical properties of the tissue water, then we have parameters that we can control experimentally in our acquisition. We can pick the pulse sequence, so is it a spin echo versus a gradient echo, which makes us more sensitive to the T2 star if it's gradient echo. We can control the TE and TR to make us sensitive to T1 or T2. And then now we're adding to that the B value. So on top of having these contrasts determined by this part of our acquisition parameters, we now have the B value that we're going to experimentally control to tell us something about the diffusion coefficient. So to describe how the diffusion preparation works, we're going to recall from those basic MRI lectures that occurred way early in the summer, that when you first put an object here, I've got this body laying down in the scanner in the bore of the magnet, and once you put them in the magnet, then presumably the water molecules are resonating at the Larmor frequency. So here we're calling that the omega 0, right? So at Larmor. And when we turn on a gradient, what happens is that the frequency of the precession frequency is dependent on the position. So as you go to a more positive position in Z, if this is a Z gradient, then the net field strength that is felt by the water molecules here is greater, where on the other side, due to the gradient, if you go into the negative Z position, then the net field strength is lower, and so the precession frequency changes. And that's given by this equation here. So the frequency is given by the main magnetic field plus the position times the gradient strength. So a stronger gradient, the greater this difference is. So now we're going to use this to do our diffusion weighting. So here's a very basic diffusion preparation, the pulse sequence. So after our 90-degree excitation, all the little protons on the water molecule have now rotated into the transverse plane. And so they're all nicely aligned here. They're all nice and coherent. And that gives us a large net magnetization, and that's the signal that we dance around to make our image. Oops, it went on without me. Gave you a sneak peek. Okay. So now if we turn a diffusion, a gradient on in X, then what will happen? Where is the mouse now? Oh. It's a very tricky, goes off of this screen and onto that screen. Okay, so if we have our gradient on that we apply in X, then these protons will feel a slightly higher field, where these will feel a lower field, so these will start to precess more quickly, these will precess more slowly, and after some time, when we turn it off, we will have what we like to call a phase twist. So there's a different phase on these protons. So these have gone a little faster, these have gone a little slower. So if we had, to help you visualize this, if we had done this in the Z direction, then we'd get something that looks like this phase twist, where each one of these is a different proton. So now that we've set this up, this kind of spatial position, then we let the molecules do their thing, have some time for diffusion, and then we put on an equal and opposite gradient. So now these that were processing a little faster last time, this time are going to process a little slower, and vice versa. And if they didn't move in their position, then they will exactly come back to where they were, and we will completely regain our signal. So that's if they didn't move. Now, if the water molecules did move, and here we're talking about if they diffused, then the effect of the second gradient will not exactly undo the first one. And so we will have lost some of that coherence, and our net signal is lower. So that's a very kind of basic idea. Does that make sense to everybody a little bit? Okay. So now, to help apply this, get it solidified in your brain, I've got two objects here. This one has water, just water in the big ball, and this one has a substance that's more like honey in it. So this is without diffusion weighting. You can see their intensity is very similar. So now if we add diffusion weighting, which one will be brighter? Will it be the water, or will it be the one with honey? Honey-like substance, I should say. Which one? Who thinks that the ball with just water will be brighter? Who thinks that the ball with more of a viscous solution will be brighter? All right, we've got one for the viscous solution, and that is correct. So when you add the diffusion weighting, if they can move more, then you end up with less signal. Okay, so typically when we do our diffusion imaging, we use a spin echo pulse sequence such that we're not losing signal due to the T2 star decay. We want to keep as much signal as possible. We just want to be sensitive to the diffusion as much as possible. So we have our 90-degree pulse, and then these are our two diffusion gradients. Due to the 180, this one has the opposite polarity. And then our B value is determined, how sensitive we are to the diffusion, is determined by the gradient strength and duration, as well as this diffusion time. And this was first presented by Stechkel and Tanner back in 1965. So here's the equation for the B value, and you can see that the B is directly proportional to the square of the length of the diffusion gradient, and as well as the strength, and then just proportional to the diffusion time as well. Okay, so now the signal that we get in diffusion imaging is equal to the signal we would have had, so our T2 weighted image, just basic signal intensity, now multiplied by e to the minus bd, b being the B value, which is what we control, and d being the diffusion coefficient that we want to find for our tissue. So for a typical diffusion weighted imaging experiment in a clinical scanner like we have over in the NMR Center, we typically use single shot spin echo echoplanar imaging, and these are reasonable type of parameters, typical parameters. So our TE, we're talking about on the length of 50 to 100 milliseconds, depending on what the B value is that you want to apply. So this is a T2 weighted image, basically, because in addition to the longer TE, then our TR is also long, so typically greater than 5 seconds. We want a fully relaxed signal because we're already reducing the signal from the diffusion weighting, so we want to start with maximum. A matrix size of, let's say, 96 by 96, that'll give us a 2.5 millimeter voxel for fields of view that are relevant for the brain. Slice thickness would also be 2.5 millimeters. We want to have equal dimensions with our in-plane resolution as well as the through plane in the slice dimension. And B values for brain is around 1,000. So now let's see, how do we calculate the diffusion coefficient? So here is, again, a ball of water, and this has very little diffusion weighting. So this is just our T2 weighted image. And then as we increase the B value, you can see that the signal starts to go down. So now we can take one of these voxels and plot the signal decay as a function of the B value. So signal as a function of B value. We can then fit our equation. We know what the signal was without diffusion weighting. We know what the B values are that we applied and how the signal changed based on those B values. So then we can calculate the diffusion coefficient on a voxel by voxel basis and make a map here of what the diffusion coefficient is for this bottle of water. So we can do the same kind of thing for the brain. So here is a non-diffusion weighted image. So this is our T2 weighted image. Now, in this image, we've kept all the imaging parameters exactly the same, TE, TR, all of that. And now we've added this B value by adding those two gradients. And we've done a B value of, as I said, for brain about 1,000. And here we've applied it in the Z direction. And one of the things that you notice probably compared to the bottle of water, so here everything in the object is decreasing at the same rate, basically. So in the brain, we have CSF, we have white matter, we have gray matter, and if you look at after we've done the diffusion weighting, there is a difference in how much the CSF has decayed compared to the tissue. And then when we calculate the diffusion coefficient, over here we can see that the CSF has a higher diffusion coefficient. In addition to that, you'll see that because we did diffusion weighting in the z direction, that there are some bits of white matter, like the corpus callosum here, that actually have less attenuation when than other parts of the white matter. And then when we look at the diffusion coefficient, you can see that it's less, and that is due to the structure. So we're going to talk about that more. But you can see basically that what I'd like to point out here is that not all of the white matter has the same intensity in the image or has the same diffusion coefficient when we're just applying in this z direction. And here we can calculate just in the same way as we did for the ball of water. So why is there this difference? Well, let's look a little more closely at the tissue. So this is an electron micrograph of a mouse corpus callosum, and this is a cross-section. So what you have here is a cross-section of axons, and the black around the axon is actually the wrapping of the myelin sheath. And so these axons are running in and out of the screen. So this shows that the water is not free diffusion in tissue, that it runs into cell membranes, it runs into myelin, the organelles within the cells, the extracellular matrix. And so the water molecules, there's some that are inside the axons and some that are out here in the extracellular matrix, and that if you imagine being a water molecule within an axon, that you'll more quickly run into the myelin sheath if you go in the perpendicular direction, in this direction, compared to along the axon. So in that way, the water diffusion is not isotropic. In other words, it's anisotropic, which means that the diffusion perpendicular is much, much less than the diffusion parallel. So it has a preference in the direction. So with this being the case, if we would like to get what the average diffusion coefficient is, then we need to be able to acquire data in multiple directions and get rid of this directionality and average it out if we want to just know what is this kind of mean for the tissue. So if we want to get the ADC, or apparent diffusion coefficient, then you need to acquire at least 4 images. So this would be the non-diffusion weighted one, and then diffusion weighting in 3 orthogonal directions in X, Y, and Z. And so you can look again at this corpus callosum. When the diffusion weighting is in X, the signal has decreased a lot because we're saying how quickly can the water move in this direction, and that is the preferred direction. Compared to Y, you can see that in the center here it's bright because it can't move very much in that direction due to due to where the axons are running, and same in the Z, the whole corpus callosum is bright. But when you average them together and calculate what is the apparent diffusion coefficient, you can see that the brain is more or less homogeneous in the parenchyma for the apparent diffusion coefficient, and the CSF is bright because it is much higher. So this is what is used clinically very often, is this kind of apparent diffusion coefficient, looking at the average weighting. And often there's the non-diffusion weighted image, and then this is what is called the isotropic diffusion weighted image. So when you take those different directions and make what is that average intensity. So here this is the isotropic diffusion weighted image at a b-value of about 720. And so you can see this area of stroke shows up very brightly. And then you can still calculate the diffusion coefficient. And if we look at what is the tissue ADC on the contralateral side of the stroke, we get a value that's typical for brain, about 0.75 times 10 to the minus 3, where inside the area of stroke, the diffusion coefficient is reduced to 0.43. So now, what if we would like to extract some of that information about the directionality and not just average it out? Well, again, recall that the diffusion coefficient in the corpus callosum in white matter, for example, is anisotropic, meaning that it can more freely diffuse along those axons than perpendicular to them. So if I have a group of these axons, I've got a little cartoon here, and I start with an ensemble of water molecules in the center, and I let them diffuse for some time. If it was free, we'd end up with a sphere, but since it's not free, and there's this preferential direction, what we get is a distribution that looks more like this. And so this is more of an ellipsoid shape. So this was, and that can be described by the diffusion tensor, which is a 3x3 matrix. And this was first published by Basser et al. In 1994. And so this diffusion tensor, which is this matrix, which describes the diffusion tensor, is 3x3, and it represents the ellipsoid. And this diffusion tensor, the matrix is shown to be symmetric, so that means we need to acquire at least 6 measurements to be able to fill in the whole matrix there. So that means the minimum amount of data we need to do diffusion tensor imaging is 1 non-diffusion weighted image, and then data with at least 6 directions at the same b-value. And these directions need to be non-collinear, such that you're fully sampling that ellipsoid. So once we've acquired the data, then we can fill in the diffusion tensor, and on a voxel by voxel basis, just as before. And so the values along the diagonal, we have the diffusion, what is the diffusion coefficient in the x direction, in the y direction, and in the z direction. And then these off-diagonal elements are not what is the diffusion coefficient in x and y, it's actually what is the correlation between the diffusion coefficient, the diffusion in x and y. So we've got these correlations between x and y, x and z, and y and z. And then once we've got the diffusion tensor filled in, we can then diagonalize the tensor and calculate the eigenvalues and eigenvectors. And this is, you can think about this as basically taking the frame of reference into that of the ellipsoid. So now the eigenvectors give us the long axis of the ellipsoid, and then the 2 perpendicular axis to describe that cross-section of the ellipsoid. And then the eigenvectors are giving us those directions. So how is that ellipsoid oriented in space? Where the eigenvalues, eigenvalue 1, 2, and 3, eigenvalue 1 goes with the eigenvector 1, and that tells us the length of the ellipsoid in that dimension. So now that we've got these parameters, we can calculate some of the things that people are very familiar with for diffusion imaging. So we can calculate, for example, the average diffusivity or mean diffusivity, which is the trace of this matrix divided by 3. So that gets us what's the average in x, y, and z. And then you can also calculate the fractional anisotropy, which tells us how different is effectively how different is the diffusion in these three directions of the ellipsoid, 3 dimensions of the ellipsoid. So the fractional anisotropy runs from 0 being a sphere to 1 being more of a pencil-like shape. And now if we look at an image of the fractional anisotropy, here the same slice, these are axial slices through the brain, so I have the mean diffusion coefficient. So this, more or less looks like the apparent diffusion coefficient because we've averaged out the directionality. And as I had said before, the white and gray matter have very similar diffusion coefficients, so you get a very homogeneous look to the brain. Where in the fractional anisotropy, now you can start to see the difference between the white matter and the gray matter, because the white matter has a much higher fractional anisotropy, where the gray matter has a much lower fractional anisotropy, because it doesn't have the same kind of coherent structure. So in addition to calculating and giving a parameter for how anisotropic is the diffusion, we can also start to try to get some information about how is this ellipsoid oriented in space. So we can do what's called directional encoding for the DTI. So if this is the ellipsoid that I've measured and I've got my primary eigenvector pointed along x, Well, now I can give a color to each one of these directions, to x, y, and z. And I can say for this ellipsoid, in this voxel, the primary eigenvector is pointed along x, so I'm going to give it the color red. Just something very, very basic so we can start to see how are these ellipsoids oriented compared to just what is their shape, which was what the fractional anisotropy gave us. So here is the same slice, but now instead of fractional anisotropy, it's the directionally encoded color map. And you can see that the corpus callosum has a preferential color in red, so running in the x direction. And then we have some running in y, so anterior, posterior, and then this is in blue running in and out of the screen. So those are just a couple of representations, but the diffusion tensor, that information can be used in other ways. These are the three that you see the most: the mean diffusion coefficient, the fractional anisotropy, and the directionally encoded color map. But we can also represent this directionality without symmetry. So here I've got positive x and negative x are all the same color, and if we go with no symmetry in that, we can start to see the difference between positive x and negative x. Or I'm sorry, this would be positive y and negative y here. In addition, we can have a linear field representation. So take each voxel and say, instead of giving a color to where that primary eigenvector is pointing, just put a line there. Just literally put a line to represent that eigenvector and make a whole map of them. So that's this line field. And you can see in the corpus callosum again, the lines are running in the x direction. We can also make maps to talk about a little bit more about the anisotropy. Here the linear anisotropy says how much is the voxel, the ellipsoid, like a pencil. So is it very, very linear to where eigenvalue 1 is much greater than 2 and 3? Compared to, we can also look and say, is it more like a pancake? Is it planar? So in this case, this would be 1 and 2 are very similar and 3 is much smaller compared to them. Or is it more spherical? So I've got a list of some of the applications people have used for DTI. I think one of the things that you hear about a lot is connectivity, trying to connect two different areas of the brain and say something about how strong that connection is. So we'll talk about that just a little bit more. But I wanted to give you more of a clinical example first. So here I have some images from a pediatric diffuse intrinsic pontine glioma patient. So for those who are used to looking at images of the brain, you might be able to tell that the pons here is quite enlarged compared to a healthy brain. So this is just the T2 weighted image, so that's about all you can tell from there. Now if we look at the mean diffusion coefficient, is it on there? Yeah, okay. Then you can start to see that there is some heterogeneity to the tumor here, because there are areas where there's a greater diffusion coefficient and areas where there's less. And then the fractional anisotropy and the directionally encoded starts to tell us that, okay, there are still some white matter tracks that are still intact, just maybe displaced. And if you compare this to a healthy brainstem in the pons area, in case you're not used to looking at them, you can see that some of these fibers may still be intact, just displaced. So we get more and more information from the diffusion compared to just the anatomical. Okay, so now one more game. Since I've been talking so much, let's give you another chance to try to think about what you've just learned. So here's this an electron micrograph again of the corpus callosum. So which ellipsoid best represents this one? Would it be the green sphere or the red, more football shape? Who says red? Okay, who says green? Very good. It is the red football shape for the long axons in the corpus callosum. Okay, so now here's an electro micrograph of gray matter. So we have lots of different types of cell bodies and extracellular matrix. So who thinks the best representation of this will be the football-shaped ellipsoid? That's red. Okay, who thinks it will be the spherical one? That's green. Yay, very good. Okay, enough fun. So what are some of the practical problems and limitations for diffusion imaging for when you go to collect this data and do your own studies? These are things to be aware of that are, some are well known and some are overlooked, but we want to make sure that you've seen them all. That way when you go to collect data and look at the data, you know some of the things to look for. So a lot of this arises from the fact that in diffusion-weighted imaging for in vivo clinical scanners, we often use a single-shot echoplanar imaging sequence. The benefits of this, the pros of this, is that it's very time efficient. You can get an entire volume of the brain in about 5 seconds. And it's insensitive to small bulk motion. And since we are trying to measure the random translational motion of the water molecules, we don't want to have errors due to, the whole head moving, if possible. Some of the cons, though, is that it is inherently low resolution because it's single shot. So that means all the data we collect for a slice has to be after just one excitation. So we don't have very much time to gain in resolution. In addition, there are distortions due to field inhomogeneities near areas of susceptibility, otherwise known as the EPI related distortions. And there's also distortions due to the actual diffusion weighting process, using those large gradients to do the diffusion weighting. So we'll kind of step through these. So the first one being the distortions from the field inhomogeneities. Here I have a axial slice from a T2 weighted fast spin echo image, so there's no distortions on this slice here. Now when we look at our non-diffusion weighted or B0 image, you can see that if I take this outline of the brain and copy it onto here, that the brain has been squished back here. So this is the EPI related distortion. This can be corrected though in post-processing with software, and we can regain the brain shape. So that's the good news about this one. In addition to the EPI-related distortions, we get distortions from the diffusion-weighted process, from the diffusion weighting process. And here what I'm going to play is a movie of the same slice through the brain, and this is before correction. There it goes. And you'll see that the signal intensity changes due to having different diffusion directions, so the diffusion weighting on different gradient axes. But in addition, the brain shape changes, and this is due to the eddy current. So when we do the diffusion weighting on different axes, then the eddy currents that are residual will actually change the brain shape a little bit as they bleed over into that EPI readout. So I'll just play it one more time so you can try to see the brain shape, kind of the brain wobble. Now we can correct for these also in post-processing software. And in that case, after correction, you can see that although the signal intensity inside is changing due to being sensitive to the diffusion in different directions, the outside shape of the brain holds nice and steady. So we were able to correct for that brain shape mismatch. Now you might think to yourself, does this really matter? Well, when we go to calculate the fractional anisotropy, for example, if you haven't corrected for that, then areas where you should have low anisotropy, like the gray matter, the anisotropy will be higher, and where you have high anisotropy, like the white matter, the anisotropy will be actually calculated lower. And so after correction, we get this, you can see that the gray matter is all nice and low fractional anisotropy. Much better. Okay, so here are some other common problems in DTI. One is low SNR because we are reducing our signal intensity from the diffusion weighting process. We're always running into that. Another one is incomplete fat suppression. There is bulk movement. If the head moves enough, then even single-shot EPI can't help you, so I'll go through that. And then also cardiac pulsation. So the brain actually moves a little bit non-linearly in the middle of the brain due to the actual cardiac cycle. So I'll give you some examples of these. But one thing I'd like to point out is that low SNR and incomplete fat suppression are two things that you can't correct for after you've collected the data. So these are things you need to be aware of when you go to set up your experiment and your acquisition parameters, that you're not bothered by these. So how about the low signal to noise ratio? Let's go through that a little bit. A common way that this occurs is people want to push the resolution and get to higher and higher resolution to have more coherent tissue within those voxels, get new anatomical structures that are elucidated from the higher resolution. So here I've got the same slice through the brain, and now I've got a 2.5 millimeter isotropic voxel, so 2.5 millimeters in all dimensions, 1.7 millimeters, and down to 1.3 millimeters. So for this non-diffusion weighted image, you'd say, oh, the SNR, the signal to noise ratio looks pretty good here. Now when we add the diffusion weighting, you can start to see that at 2.5 millimeters, when you look at the corpus callosum, let's say in this part where we actually have a lot of signal loss due to the diffusion weighting, it's still well above the noise level. When we get down to 1.7, it's pretty close. This is just from visual inspection. And when you get down to 1.3 millimeters, it's hard to tell them apart. So this is very low SNR. And just to give you an idea, when the voxel volume, it's the cube. So at 2.5 millimeters, this is our voxel volume, over 15 millimeters cubed. And when you get down to 1.3, although that's not quite half in one of the dimensions, the voxel volume is all the way down to 2.197. So it's significantly reduced as you drop your resolution. Now, does that low SNR matter? Yes. And when we look here at the fractional anisotropy maps, you'll see that if you've got very noisy images, then again the things that should have low fractional anisotropy get calculated higher, and those that have higher they get calculated lower. So it does affect the quantitative parameters that you want to extract robustly from your data. So don't push the resolution. Stay out of the low SNR regime. And here's another artifact that I'd like to make you aware of that is very common that people don't always notice or don't recognize, is this is an incomplete fat suppression. So right here is actually what happens is that the protons on fat resonate at a slightly different frequency than those of water. So in single shot EPI, what that means is that you get a fat image that is displaced compared to the water image. And so this is unsuppressed fat that is right on top of the brain. And because the fat has a very low diffusion coefficient, when you have diffusion weighting, then the signal is still quite bright. And then this actually, the signal here then for the brain is incorrect because it's contaminated by the fat. So when you look at the mean diffusion coefficient, then you calculate something that's much lower than the tissue, and it also affects the fractional anisotropy where it is also lower than it should be. So it's important to look for these kinds of artifacts, and if you see it in the data that you're collecting, then you got to stop the scanner and try again and see if you can get a better fat suppression. And this is just also to make you aware, there's a cardiac pulsation effect. So this is an axial slice, and what happens is when the heart beats, there's a bulk of blood that comes into the head, the skull being a fixed volume. As the blood comes in, part of the tissue comes out, and it's like the spinal cord's kind of getting pulled. So all the stuff in the brainstem and the midbrain get pulled down, and so there's a shearing of the tissue there. And if we have diffusion weighting in that direction, when that part of the cardiac cycle occurs, then you get a drop in the signal that's not due to that random translational motion that we want, but more so due to the tissue shearing. And so there are ways to estimate the tensor in a robust manner to not have these affect your calculation, or you can also gate to the cardiac cycle and collect data that doesn't have this error in it. And then another example that is probably one of the most common things seen is bulk movement. So if there's a subject in there that is really moving their head, then what happens if you have a rotation, when the diffusion gradients are on, then that causes a shift in the case space. And if the shift is big enough, then the energy gets outside your acquisition window, and we start to get these slices of just noise. So here's one where you have like half the slice noise, and if I take all of these axial slices and resample them into the resample them into the sagittal plane, then you can see that I've got multiple slices of dropout. So clearly you would not want to use this volume in your calculation because the signal loss here is not due to diffusion, it's due to this greater bulk motion. So we just have to throw these whole volumes away when you do your calculation. Okay, so I also wanted to touch just a little bit about beyond the diffusion tensor. So most people probably, when they think of diffusion, might think of tractography. So what is tractography? It is the use of this orientational information from diffusion imaging to reconstruct estimates of white matter pathways in the brain. So you use, like I said, use that the distribution that is calculated, and then you start to connect the dots effectively from voxel to voxel and make these tracks. It's one of the hottest utilities. So for diffusion tensor, there's a limitation, and it comes from the partial volume effects. So here's the brain, and so for a typical resolution being 2.5 millimeters, I put that little voxel here. So you can see there's a number of bundles that run through that, and that's just at this level of the naked eye. So at microscopic, there's even more. And so this partial volume effect comes from that if we have all of our axons, let's say my little cartoon here, running in the same direction, then our distribution of the water from motion will look something like that. And so the diffusion tensor ellipsoid well represents this. But if we had two different groups of axons that are running, let's say, in perpendicular directions, then what the distribution looks like is more like this, more like the anatomy, but what we would calculate in the ellipsoid is more of a pancake shape, so it does not represent that as well. So one thing one can do is reduce the resolution, and I just wanted to show this image here of a sub-millimeter DTI to where if you get the voxels very, very low, then you can start to recover some of those things that are lost from the partial volume effects. But this is very slow. But what people typically will like to do is not represent it with the diffusion tensor. So they'll use high angular resolution diffusion imaging. They can either represent it with multiple tensors or some non-parametric algorithms like diffusion spectrum imaging or Q-ball or spherical deconvolution. And what those algorithms do is instead of calculating the ellipsoid, they'll calculate the fiber orientation distribution function like this or like this in this case. And so from there, then they'll try to track both of these through both of these. So one of the things about that type of data is there's nothing for free. So if you want to do some of these algorithms, then you have to acquire many, many more volumes of data. And even with that, there's still quite a bit of literature out there, even more recently, that shows some of the issues with the inaccuracies of tractography. So with that, I'll like to thank Dr. Bandettini for inviting me to participate in the course, Dr. Pierpoli, who's your next speaker, for giving a number of the information in the slides that I presented, and some other colleagues who also contributed to the slides. Of course, I'd like to thank you for listening, and I'm happy to take any questions that have come up as we went along. Any questions? Sure. Yes.
For example, once you want to analyze just mean fractional activity, relate that to the volume measure in the brain. And is it also, should you also consider partial volume effects? Like, for example, if you find an association between mean FA and volume in the brain, can that association be just due to partial volume effect rather than any pathophysiological reason?
So I'm going to repeat the question just because they're recording it so that we can, whoever listens to it in the future can. So I think the question was, so as I mentioned partial volume effects as far as white matter running in different directions, but you're asking what about partial volume effects as far as like the CSF in the brain tissue? Is that correct? No.
Not CSF, just mean FA, for example, in certain regions of the brain.
Uh-huh.
Yes,
that is true. So what about partial volume effects for just the fractional anisotropy?
Yeah.
In general, yes, absolutely. And that's something I should have pointed out, but now that I think about it, I did not. So if we look at one of, if I go, let me find that slide. So here, for example, how do I get there? Okay, so here the like the mean diffusion coefficient is quite consistent, and here this is all healthy white matter, right? But the fractional anisotropy is very low here compared to right next door, and the reason why the fractional anisotropy low there is just what you were asking about, and is the fact that there is white matter running in more than one direction. So within that voxel, it looks isotropic because of the different directions that the axons are running in. So what we measure is something that's spherical, but the underlying tissue has axons running in multiple directions there. So this is also a partial volume effect, yes. Yeah. Any other questions? Okay, well, thank you for bringing that up, because that's something I definitely meant to point out earlier. So, okay, and with that, I'll end. Maybe we'll give you guys a 10-minute, 5-minute break, and then we'll pick back up with Dr. Pierpoli. Thanks.